take this opportunity. So I wanted to take this this opportunity to first reflect on 20 years of a scientific advisory board that really deserves the acknowledgement and the recognition. So for the last 20 years, we have worked with this wonderful group whose pictures you see here, and I had the honor of serving as the founding chairman for this group. A lot of things happened as a result of collaboration, of putting basic scientists, translational and population scientists, and clinicians together to share ideas, and mostly driven with a lot of collaboration and a lot of initiative and guidance by these two wonderful ladies that you see here. And together, this group went on to formulate the guidelines for evidence-based management in our disease, to initiate the idea of creating centers of excellence that bring together clinicians and researchers studying the disease, and clinical centers all aimed on deploying the guidelines, so that, you know, when you go to a place, you get the recommended type of MRI, you get the recommended clinical information that follows the best guidelines, rather than walking into a place and having your doctor look up CCM on Wikipedia and advise you. So I think this movement toward the centers of excellence and clinical centers played out and has been an amazing change in the field. And it required a lot of time of various members of the advisory board, interviewing and nudging on and helping these centers organize and come together. This group went on to share with the patient community the interpretation of novel scientific discoveries. What does it mean when Brent Derry describes zinc in the worm? Does that mean I should go eat worms or eat zinc, or what do I do, you know? Or when there is a signal that we see in important clinical patient analyses by Dr. Fleming on birth control pills and so forth. These have been very important, and webinars were done. And I think this group has done a phenomenal job. And last but not least is the response to the COVID pandemic that changed everyone's life, including those with cavernomas, and everybody was looking for guidance. So I get a vaccine. Is it better to get COVID or to get a vaccine? And what is going on? And if I have a brainstem lesion, does that matter? And so forth. So it's been really wonderful, and also it's nudged us to think about things that we haven't really been as good at in this group, but we should be. Things like how do we get the knowledge to the racially and economically underrepresented groups in our midst. So we're not only serving the suburban white population around the country, but really we're reaching out to everyone who has CCM. And that is, as a result of 20 years of this, the organization was reborn and renamed. And as you know, this year is a chance to change the name with a real commitment to be an alliance to cure. And I think this is a wonderful new goal and new mission ahead of us. And I think because of the tasks at hand, the reorganization of the advisory boards, you heard a little bit about it yesterday. And on the clinical side, a wonderful team of clinicians who really are the leaders at our centers of excellence and clinical centers and clinical researchers, will be led by Ed Smith and with a designated associate who will be the successor after Ed's service. So we create a continuity. So we are really wonderfully blessed to have Kelly Fleming having agreed to do this with Ed. And on the scientific side, a lot of discoveries have gone on, and actually it is a wonderful opportunity to have these pair of characters that you see on the right side, Doug Marchuk, who I will speak in a couple of minutes about, who will start by leading the scientific advisory board and having Brent Derry help him and move on in a few years into his role. So I want to just give a hand to our new board leadership. So people will ask, well, what the hell are you gonna do? You know, so not to worry. Connie and I negotiated that I'm very, very happy to remain as an emeritus member of both groups, with the main idea of bringing ideas back and forth between the science and the clinic, and continue to work with both groups to whatever extent they want as we continue to move the field forward. So this is a very good chance for me to move to the main task of my presentation this morning, which is to tell you a little bit about Doug Marchuk. So Doug may not remember this. The reason we're dressed like this is because my wife kicked us out, so we smoked the cigars outside, and it was very cold. What this picture illustrates is really something that is very unique about Dr. Marchuk. He and I started in the 1990s as competitors in a big way. I was working with the team at Yale on looking for the gene for cavernoma, and we discovered the common Hispanic mutation, etc. While he was also on the same quest, and it was which paper was going to come up in what month of the same year type of competition between the groups. And then within 2 to 3 years of this type of typical healthy competition in the field, actually Doug reached out to me, and he said that, I remember, he may not remember, but he told me that this field is about genotype and phenotype reflecting each other. And my life's work and the way I got tenure at Yale was working on the phenotype, which is what does the lesion look like? How is an AVM different from a cavernoma? And he is a genotype guy, and why should we compete but not work with each other? And it was the time that the ideas came to, since we knew the gene, to try to create mouse models and take advantage of imaging the mice and looking at the lesions in the mice. And there were some wonderful times when the first picture of a cavernous malformation in the mouse brain was there, and Robert Shankar remembers this very well because we did the histology in the lab at Northwestern, and I couldn't wait to call Doug and tell him about it. And several big NIH grants later, we remain collaborators, but this paradigm of competition turned into collaboration is really what this whole alliance is about and motivates this alliance. I want to tell you a couple of other things about Doug that I learned other than this competition collaboration theme. He is truly a man of virtue. He is generous, he is kind, he pursues justice to everyone who is around him, whether they're people in his lab who have gone on to have wonderful careers. Amy is but one example of them. And he has prudence like you wouldn't believe. He's a very prudent guy. Whenever there is something going on that doesn't make sense or whatever, he takes time. He doesn't respond too quickly. And then eventually he has some ideas about how to sort it through. So he has really inspired me to be a better person because of those virtues. So yet this humility and openness that he has belies an amazing pedigree. This is a man who started and has remained at the highest level of distinction in the American Academy. Doug, while he was eating fried chicken, he was also doing a PhD at the University of Chicago with Elaine Fuchs, and looked at stem cells and the genes that make a skin cell become good or bad before any one of us were thinking about looking at any of this. And after his PhD at the University of Chicago, he didn't think this was elite enough, so he joined the big laboratory at the University of Michigan of Francis Collins, of all people, who basically taught him about looking for genes that motivate disease. And as a result of that, of course, we know that Dr. Collins went on to head the National Institute of Health and to map the human genome. But one of his truly distinguished trainees is Doug Marchuk. And then Doug went to Duke and started working on diseases from what he has learned from Francis Collins. And he made incredible contributions beyond cavernous malformations. So his footprint is in the area of neurofibromatosis, genes of hereditary hemorrhagic telangiectasia, somatic mutations causing Sturge-Weber disease. And the part about CCM is the one I'm not going to tell you about. One, because you know all about it, but second, because it's going to really be his keynote address theme to tell you about his life journey and odyssey with CCM. And he has risen through the ranks at Duke University to earn the title of the founder of the university. To be a James Duke Distinguished Professor at Duke University is the ultimate recognition. So we are, as a group who have had a chance to walk with him, part of this journey, and to have advantage of his leadership and contributions, are particularly happy and honored and proud today to introduce him to deliver our keynote address.
So, you know, as you know, this man has been by my side now for 20 years, just moving this field forward in ways that we could not have imagined. And so to mark his promotion to emeritus status, we made him something, and he's going to have to sit there and get a load of what we made for him and then be able to take it home in its short and uncut version. So enjoy.
Who would have guessed 20 years ago that the simple email that you received from a mother in Virginia that you responded to so quickly would have led to all of this? We have worked together side by side through good times and sometimes not so good times to create something of value, to engage together in what you call a marathon of love, to help our patients, our families, and to work together toward a cure. And we could not have gotten this far without you by any measure. I am eternally grateful.
Hi, Dr. Awad. Thank you for all that you've done for me and the Keynote community. I hope to see you soon. And you're the only doctor that I really liked going to when I was younger.
Dr. Awad, we were lucky enough to meet you for the first time last night, and you've already had such a big impact on our son and our family. So a big thank you from London.
Hi, Dr. Awad. Still wearing my Awad Squad shirt. The morning of my surgery was during the pandemic, and I couldn't have anybody with me, but you took my hands in your hands and let me know that I was never going to be alone. You were there with me. We are so fortunate that your compassion is just as extreme as your brilliance. Thank you for saving my life.
I'd like to take a second to thank Dr. Awad and his team for the kindness and compassion they showed to me and my family.
Thank you for listening to us and including us, demystifying CCM, and your never-ending commitment to research, treatment, and a cure.
Dr. Awad, thank you so much for all you've done for the CCM community. We wouldn't be here today without your help.
Hi, Dr. Awad, it's Tony Mayer. Thank you for your 20 years of service to the Scientific Advisory Board. We are so grateful to have you as part of our organization, and a special thank you for all that you've done for my family as well. Cheers, my friend.
Yeah.
Thank you, Dr. Awad.
Dr. Awad, I'm happy to have the opportunity to thank you for all that you've done for me and for my family. You've allowed me to keep working, my daughter to go to college and thrive, and your research has given us all hope for a cure in the future. Thank you so much. I can't tell you enough.
Dr. Awad, I'd really like to thank you for your many years of service to the Alliance.
Hi, Dr. Awad. We just wanted to add our appreciation, our gratitude for everything that you've done for the CCM community. Most of all, thanks for what you've done for our family.
Thank you. I love you.
From Cavanoma Alliance UK for everything that you have done for Cavanoma, not least from our perspective in talking to us on many occasions. Thank you.
Dr. Awad, my name is Kristen Meek and I'm from Vero Beach, Florida. And I found out as a little girl I had cavernous malformations. And I just want to say thank you so much for the work that you do. It's so empowering to know that someone's out there who is looking to help us find answers.
Hi, George from Brazil. Ethan Awad was the right man at the right time and right place. His striking knowledge helped us to understand cavernous malformation. He's a good friend.
Dr. Awad, thank you for everything that you've done for the CCM community. From the research to your patient care to how you help millions of people around the world and the future of the CCM community. We cannot thank you enough. You truly are one in a million.
Hi, Dr. Awad. Thank you for being our scientific advisor, for all the webinars, all the information, and for giving me hope that someday we might have a cure. Thank you.
Thank you so much for everything you've done for me, Dr. Awad. You don't even know how much you mean to me and how grateful I am to you and how grateful all the members of this community are to you. You've saved so many of us and you've advanced the science behind cavernous malformations in a way that nobody else has. Congratulations on 20 years. Thank you so much.
Hi, Dr. Awad, it's Shelby Bradshaw. Wanted to say thank you for all the help you've given me and my family. You've put my mind at ease as someone with the disease, and you have given me hope for the future. So thank you for everything.
Dr. Awad, our entire family thanks you for your passionate work and dedication in finding a cure for this disease. Thank you.
Thank you, Dr. Awad.
And just thank you, Dr. Awad, for your 20 years of service.
I am so glad to be rid of that crappy CCM that caused my midline shift. And Dr. Awad, you and your team are amazing, and I feel very blessed to have had you to rely on. Thank you, thank you, thank you.
Hi, Dr. Awad. Thank you for always having an answer to the questions or taking the time to research the questions that we have for you. I appreciate your expertise, and I'm always grateful to you and your team.
Hello, Dr. Awad. It's Camden here. Thank you for being my neurosurgeon, and thank you for everything you do.
I'd like to say thank you for all the work that you have done. You are such a great person.
Hi, Dr. Awad. Nicole Clayton, representing the whole Clayton crew, CCM3. Thank you so much for everything that you've done for the world of academia, for the scientific community, and especially for the community of people with cavernous malformations. I cannot thank you enough. You are a life changer and a difference maker.
Dr. Awad, congratulations on the 20-year anniversary of working with the Alliance to Cure Cavernous Malformation. I find myself in the wonderful position of being a patient of yours since April of this year, and it gives me tremendous relief to be under your care, and I can't wait to see where your research goes in coming up with a cure for this condition. Congrats again.
All right, so what we did was we created, first off, thank you to Darla for putting that together, collecting the videos and editing it, and also getting this really cool USB stick. That was 6 minutes of patients saying thank you, as well as a few other notable people. There's 16 minutes of full, complete coverage here with patients that we couldn't fit into this. So that is just a fraction of the people that Dr. Awad has already touched, and I am sure there's going to be almost as many more still to come. So thank you, and Doug, it's your turn now.
You know, well, there's sometimes they talk about a tough act to follow. One thing you can say is that it's now taking 4 people to do Issam's job. If you noticed, the board has split into 4 people. So we may need some help, Brent, on our side. And anyway, all right. So anyways, first off, thanks, Dr. Awad, for those wonderfully kind words. And I want to say it's an honor to speak to my scientific and medical colleagues here. But it's especially an honor to speak to the patients and the families. And as you can see from the videos, everything we as a community do is for you. And so that's why it's such an honor to speak to you. But it's also terrifying because I want to give something to everyone, have something, a little bit of something for everyone. And I really struggle with what am I going to talk about in a keynote address? But when I thought about CCM research, which I've been doing for more than 20 years. I thought it was like there was an odyssey, a story there. And so that's what I'm going to do. I'm going to tell you my personal odyssey of the research. And I've titled it Maps, Genes, and Mutations, because that's where the trail led. Which one is it? Oh, so do I point it at that screen? Okay. No, I should have practiced. It is the forward button. I promise you, it's not there. Can you forward it until we maybe change a battery or something? He's doing it there. You did it? Okay. That one didn't work. I could promise you. Okay, now it does. I moved the batteries. Okay, so the first thing I got to talk to you about are maps, and I really love maps. I always get 2 copies, or I used to before GPS, of the Rand McNally Road Atlas. One goes in the car, but the other one stays in my home, and I just flip through it, and I can spend hours just amusing myself, basically, looking at places, looking at features, how to get from here to there, etc. So let's think a little bit more about what a map provides. And I want to preface this by, I was in New York in 2001, and I was walking by the New York Public Library, and I saw this exhibit, and it was called Heading West, Mapping the Territory. And it was a sort of a compendium of all these old maps as they were mapping out the West, like Lewis and Clark and others. And there was a saying that I loved, and I made a slide of it. And I actually, one of those things that we used to use when you did PowerPoint presentations, not PowerPoint, but slide presentations. And I found that old quote and I rewrote it here. According to an old adage, a place is not discovered until it's been mapped. So let's think about maps more. So this is considered the first detailed map of the world. Apparently there were others, but there's absolutely no copies of them. So this is Al-Idrisi, Moroccan geographer, and this is considered the first map of the world. How does it look to you? Can you get to anywhere? Would you be able to use this to get anywhere? It's kind of a little bit low resolution. Actually, it's much better than you might think, because we have a sort of a tradition of putting north up and south down. There's no reason whatsoever to do that. And if you flip it, it's not so bad. You can maybe start to distinguish features, okay? Well, let's think about the first map of the human genome. This is the first map. This is called a karyotype. The chromosomes aren't all lined up, but when they smash a cell, take a picture, they move the little pixels in the picture, and you can see there's 22 autosomes and then the sex chromosomes, the X and the Y. But if you said, well, find me the gene that causes, or the genes that cause CCM, how would you know where they are? So this is what you call a low resolution map. It's the equivalent of Al-Idrisi's map. Everything's there, but there's no detail or resolution. Where are all these genes? At the time that these things were going on, scientists estimate 100,000 genes. It's much less than that because the genes are split up and spliced together in different ways so that it can make many more combinations. But the point is where are they all? Where are they all? And which gene, when mutated, would cause CCM? So to do this, we have to find out where these genes are located, and we need a map. So the first thing we need to do is just find out like what chromosome it's on, okay? And there was, this is a contribution that we made with Leslie Morrison, and you can see the two other groups who were involved in this, including Gunnell et al., which is Dr. Awad and the team at Yale, and Dubovsky et al., which is the team at Prevention Genetics. And we found, independently, but, cumulatively, it was very clear that there was a gene on chromosome 7. And this is just showing a couple of families, CCM families, that Leslie had collected in New Mexico and that we had done the linkage on. And so if you look at all the data from all the groups, you could say, that gene is somewhere on chromosome 7 in that very tiny region. So, and then there's a line and it says 4 centimorgans, 4 cm. And that's the level of resolution. So we've got it between somewhere, but there's nothing there to look for. There's no map. So what are we going to do? That's basically the equivalent to saying, someone please go find my house. It's somewhere in that state, which is North Carolina, by the way, if you didn't know. But it's a black box. It's all darked out. You can't, where would you start looking? And so this was before there was even DNA sequence. Now you type in and you say, what's the sequence in between here and here? You could just pull it on your computer and everything. There was no sequence. We had to build the map. So we were mappers. We were the Lewis and Clark of that region of chromosome 7. And we teamed up with Eric Green, who at that time was at the Human Genome Institute. He's now the director of the National Human Genome Institute when Dr. Collins went to be the head of the whole NIH. And we teamed up with this person who, Dr. Awad, I think, or someone showed a picture of this is Eric Johnson, and my lab. And we started to build that map. And here's the first publication of a map of that region. I'll show you what it looks like in a minute. That's what it did look like. Remember, this is like there's 4. This is like the whole state of North Carolina, and at the end it looked like that. Now that still doesn't look like much to most of you, but what those are individual pieces of DNA, and we know how they link up together, so that we can then sequence the DNA. So not too many people know this, but these clones that I'm showing you here in that paper were the template for sequencing that part of the DNA for the Human Genome Project. You see, back then, Congress and the genome people were worried. We're spending what appeared to be a lot of money. Actually, it was a very good investment, quite frankly. And Congress was saying, oh, show us something for our money. And so the community was saying, instead of starting on chromosome 1 and going all the way across and finishing that and starting on 2, they said, let's focus on regions where the research scientists have found that somewhere in here is a disease gene. Let's get some wins right away, early wins. So I actually nominated this region. I wrote the letter and everything to the National Institutes of Health, and they chose it. and partly because Eric Green was at the NIH at the Genome Institute. And then we were putting together this map, sending clones, and they were sequencing it. And I want to point out that how did we get the DNA back? It went immediately into the public domain. It was there for anyone in the world to see. And then we just pulled it back out. So we didn't do this for ourselves. We did it as part of the Genome Project. So now where is that gene? So I'm going to tell our part of this story, and there's multiple different ways to do this, but Dr. Awad talks about this. In '96, Murad Gunel, Sam Awad, Rick Lifton, and their team noticed that there was a common fingerprint of DNA in some of the Mexican-American families, the common Hispanic families. The way we did this was we took all of our families from Leslie, Morrison, from all different places who were of Mexican-American heritage, and we started to look at that fingerprint, and we gradually narrowed where you see where the box is. The gene isn't all this here. It's got to be in here because they're probably all related. This was work done by Trilok and Sahu. And so we mapped the interval, and then we just started sequencing every gene because we knew now all the genes because we not only had the map, but now we had the map at one base resolution. We had every sequence of every gene. And we found this gene called CRIT1, and we found mutations. And in different families, we found different mutations. Those are all those nomenclature across that green bar. That's just a picture of part of the gene. The mutation in the circle was the common Hispanic mutation. It was a founder mutation, meaning that it was in all the people of that heritage because they were all distantly related. So far distantly related that in fact they wouldn't have known that they were related, maybe deep looking through medical records, but their DNA would have not told you they're related except for right around that mutation. You may know this if you have ancestry.com or 23andMe, that you go out to the third or fourth cousin or something and you share like 1%, less than 1% of the DNA, but the DNA these people shared was right by the mutation because it was ancestral. They were all related. And that's our publication on that. Sahu, who I showed you a picture before, started to, oh yes, I forgot to mention, this gene already was known. They didn't know where it mapped. They didn't know that it was on chromosome 7 until we made the map. So it was like a little village somewhere, but then we found exactly where it was. And this gene was the gene that caused the disease because we found mutations in the gene. But it was called CREV interaction trap 1, a nickname CREV. Scientists are good at making these acronyms. And there was a single solitary paper on it by Erica Galima's lab. Basically, that's how she studied it. And so she said, this is interacting with CREV or RAP1A. That's all she knew. So Sahu in my lab started to try to make the protein. And he came to me one time, he says, I can't do this. This is not working. I don't think this is the real protein. I think it's the protein, but I don't think it's the whole thing. And so what Sahu did was he started to look upstream, like the gene is here. He started to, in Durham, he started to look, well, let's look at Chapel Hill and go out to Hillsborough and keep on going. And sure enough, the gene kept on going. Erica Galima's found part of the gene, but she didn't find the whole gene and all the protein. And so using a trick, which scientists know is 5' race, and we did this is my major contribution so far. I started to look in the database. This is my dabbling in bioinformatics, using computational evidence, looking in what's called the express sequence tags and stuff. And I saw all these genes and genes, all these little regions that could have made a gene. And we thought maybe they're all put together. Maybe this is one bigger gene. And the bottom part just shows that when we got to the very end, we thought, now we're at the end of the gene because the mouse, the human, the rat, it looks identical. Did I? So what Sahu essentially found out was that there were 4 new parts of the gene. There were 207 extra amino acids missing. And I said, Sahu, you know what would really seal the deal? If we found a mutation in those new parts of the gene that we hadn't found a mutation in one of the families. And you see that in red box there, we found a mutation. I said, this seals the deal. It's the same gene, it's just part of it, okay? Little anecdote, I was at a meeting and I heard in a European country, they were doing diagnostic testing for CCM. And I didn't think we were doing it yet in this country. And then I I told this person, I'm gonna say their name. I said, do you know that there's more to the gene? So you better find the other. I told them that there are more genes because I didn't want them to say, oh, you don't have a mutation when they were missing part of the gene. So I told you, Erica Galimas had this paper. She said this CRIT protein interacts with RAP1A or CREV. And John Zawistowski, who is right there, was in my lab. They've all come back to here. Thank you, John. And John said, well, let's take this new part of the gene, plus a little more, that's kind of uncharacterized, and let's go back and go fishing in the sea of other genes and see what it interacts with. And he pulled out this gene called ICAP1, and he got 2 really nice papers on studying the biology of that. And when we looked at the sort of the sequence features of it and the features of the protein in terms that it encoded, we said, you know, at this time, we knew that there were at least two other CCM genes that were in different places in the genome. And when we looked at that thing, we said, you know, that looks like an awful lot like a predicted gene on chromosome 7 in another region that we know another CCM gene lies. So we were saying, this guy that John pulled out looks like that new gene. That gene had never been studied. It was a hypothetical gene. Here was its clever name, MGC 4607. It was the 4,607th unknown gene. You could tell how many unknown genes were at that time. And we said, gosh, they look the same. Maybe that's the second CCM gene. So Christina LaQuarrie in the lab, who was doing more of the human genetics, John was doing more of the molecular biology, she started sequencing that gene in some of our families that weren't CRIT families. And sure enough, she found mutations. This is the second gene. We tried to name it melcabernin, but it didn't stick. So in the end, that was the second gene. The third gene was found by Tournier Lesèvre and her colleagues. And there's an interesting story. I won't take time to tell you about why we missed it, but they also independently found these genes. So now we have the 3 genes, the CCM1, CCM2, and CCM3. Maybe those are better names anyways. So I want to tell you 2 more stories about ancestral founder mutations. I told you about the common Hispanic mutation, which is really an interesting story. And it's the way we eventually found the gene itself. But I want to tell you 2 more stories, one about the Ashkenazi Jewish mutation, which we found, and the ancestral colonial mutation that Connie alluded to. And the first story of the Ashkenazi Jewish mutation starts at this meeting some years ago. And Jim Weber, who's the head of prevention genetics, was doing DNA testing. He got up and he said, we're finding all these mutations. We had stopped really sequencing families because it really belonged in the clinical realm where it was done as a clinical service. But Jim said, I found this weird variant. I don't know if it's a mutation or not. He didn't have the right to call it a mutation because he couldn't tell. But he found it, I think, 7 times in different families, but they had all checked the box that they were Jewish. And he was just up. It was a little small, the room by then, but he was up there telling that story. And I thought, oh, we could try to figure this out. So what we did was, and we is Carol Gallione, who's back there. Carol, we first looked at that DNA fingerprint around what he thought might be a mutation, might not be a mutation. And sure enough, the fingerprint was the same. And all those families, and we got a couple others from various people who had the same variant. And then there's the variant. It's not in the coding region, so it's not in the region that encodes the gene, and it's not at the invariant splice site, but it's a little bit further away. It's actually 2 base change. And this is only for the scientists and the aficionados. This part's for you and not for everybody, and probably not even for all the scientists. But you really can't skip exon 1, right? Because then there's no transcript, so you can't find the mutant transcript. So we did Carol, again, did 2 tricks. I won't go through this one, but she used an alternative. She found an alternative exon and showed that it was skipped when you used the real exon 1, but not when you use the other exon. But more importantly, we made a fake exon. We stuck together an internal exon with the back end of the CCM exon 2, and it caused missplicing. So there is no transcript normally, but we made the synthetic system, and we were able to show that this was, number 1, ancestral. In other words, it was probably deep in the Jewish community, maybe way, way back, and that it was really the Jewish mutation, which is now commonly used as a sequencing. If you're Jewish, that should be the first place to look. And then the third story on the ancestral mutation is the one that Connie mentioned. And I guess I'm trying to think now, maybe the patients didn't hear that story, but when we started, we always miss mutations because sometimes the ones we sequence, we only find the ones that are simple little changes in the code, but sometimes there's big gaps missing, and you can't see that by sequencing because it's not there, so you don't sequence it. And Christina LaCourie, who I mentioned before, found these large deletions, and some of them were smaller, but these are huge. These are like deleting out all of Durham or Chapel Hill. This isn't like a mutation like the house burnt down. This is the whole thing. But she found one mutation in 8 families that showed the identical breakpoints, and she mapped it down. And they were like, how could this be identical? And we published it and we left it at that. But then Connie calls me one day and says, we keep seeing this all the time, the same mutation. This is the CCM2 exon 2 to 10 deletion, it's called. And it's pretty common, at least in the United States. So she had, like, at that time, 19 more families. So that's, this is the same mutation in all these families. And so we used a combination of genetics, which we did. And then Connie and Henrietta Christmas did genealogy. And we found that it was a common mutation. So here's the genetics part that, again, Carol Gallione did. And basically that fingerprint around the deletion, not in the deleted region, it's gone. There's a huge gap, but on just outside that, the DNA fingerprint is identical. And so we call it the same haplotype, which sounds like they're related. But then the genealogy, all these 19 plus the original 7, whatever, however many families there were, they started to coalesce into larger families that we're only showing you parts of the families here, but it looks like they were all part of one family. And like I said, Henrietta Christmas there did this. And then what Connie and Henrietta did was try to figure out, well, maybe we could figure out where this started. And it looks like it started in the 1600s in Virginia. And eventually, over the last 400 years or so, it's spread out, okay? And so this is a common mutation. And what's the value of this? It's really understanding the natural history of this disease. One thing is that it can be common and it can spread and it's compatible with life. So that's the story there. I want to then focus on something that perplexed us all along as a geneticist. Every patient who has CCM has a mutation in every cell in their body. It's a dominant, so you have one mutant copy and one normal copy, but it's there in every cell. So how does that relate to the fact that the CCM lesions are discrete and focal? There's one here, there's one here, they're in the brain. It's the whole brain vasculature isn't bad. How does that work? This is just showing that a clue came from thinking about CCM and its epidemiology. It's the way it presents. So there's inherited forms and there's sporadic forms. And generally, and you can just look on the right, generally the sporadic forms, like no family history, no apparent genetic mutation, the patients get one and only one lesion. Whereas in the autosomal dominant form, this is a particularly severe case, you can get many, many, many lesions in the brain. And that made me think all along, actually, that this sounds like retinoblastoma, which is very similar. The inherited cases, it's a tumor of the retina. The inherited cases get multiple tumors in both eyes, and they end up going blind in both eyes, these kids. The sporadic cases have one tumor, only one in one eye, and they go blind in one eye. So you see the differences. It's one versus many. And Knudsen, the late Alfred Knudsen said, he called this the 2-hit idea, that you have 2 copies of every gene. And what he was saying was, in order for the actual malformation to occur, you have to have 2 mutations, one in each copy. Now that sporadic case, that person, they would have had to have 2 mutations in the same cell happen. That's like lightning striking twice on the same person. That'll happen maybe, but very, very rare. But the familial case, the case where you've already got one copy mutated, every cell, every endothelial cell in the brain is one step away from missing both copies. And that's the 2-hit model. And this is just a picture of it, whether you have one mutation if you're inherited, but you got to get that other one to get the lesion. And so this is a case where the convergence of Dr. Awad and myself is in my lab. Amy Akers was working on this. And we all know Amy, and she's somewhere here. I missed where she was. She just spoke. Hi, Amy. And I hope you like that picture. That's her at the lab bench a couple of years ago. And so she had a strategy, and basically this is before, for the aficionados, this is before next-gen sequencing, or I would say actually just at the cusp of next-gen sequencing. And she would isolate the lesion, amplify all these GNA, sequence individual clones. If she found something, she had to do it again. If she found something, she had to do it again. In other words, she tried to prove herself wrong. And I keep finding it. Even if I don't want to find it, I find it. Then it's real, right? That's when you know it's real. And this is just some data on that. And the point being here on that top in panel A is you couldn't find this if you didn't sequence individual DNA molecules. This is the old-fashioned way of doing it, by the way, and Amy will admit, extremely painful and arduous. But if you look at the bulk lesion, if you just took the CCM and tried to sequence it, you wouldn't see the mutation. But as you sequence individual molecules, you say, oh my gosh, some are normal and some are mutant. And the mutant ones, we found the second hit. And below there, she showed that it was truly 2 hits. That is, the germline, or the mutation that was inherited, came, like, say, from the father. The mutant somatic mutation, the one that was acquired in the lesion, was on the maternal copy. So both copies were knocked out, fulfilling Dr. Knudsen's idea. And what furthermore she showed was that if she microdissected in different parts of the tissue, the mutation, the somatic mutation, was in the endothelial cells, the cells lining the vessel, and not on the outside. And again, Judy Galt, also with Dr. Awad, published this. And this is about the time when, I think actually even before this, we were working together, but the papers came out about that time. So I'm going to fast forward a number of years because I'm thinking about, now we've gone from maps to genes to mutations. And now we start to think about somatic mutations. That is, mutations that happen during development. They're not happening, or maybe when you're an adult, they're not happening. They're not in every cell in your body. You're not going to pass it on, but it's in the tissue. And we were thinking, what converts the CCMs into an aggressive state? You can see a solitary lesion on the left, in the middle, a particular familial case. Then you see another familial case with one really bad lesion. Is something going on there? Could somatic mutations be playing a role? Well, in cancer, there's this progression of somatic mutations, that things have to happen. Like, this is the famous, what's called Vogelgram, named after Bert Vogelstein, who really for years and years discovered this in colon cancer, that you need one mutation in this gene, but then, and that becomes an early adenoma, and then you have another mutation that becomes a late adenoma, but it doesn't get to be the bad boy, the carcinoma, until you get a third mutation or so. So this idea that there's a progression of somatic mutations. And in the end, they should be in the same cell, because that cell now, at least one of those cells, has to become the carcinoma because it's got all the mutations in the same cell. Now, people had been starting to think about this, about vascular malformations, because this is just a short list of some of the papers where people are finding somatic mutations in the vascular lesion. In other words, there's no family history, it's not going to be passed on, but that vascular malformation, whatever type it is, there's Serge Weber there that Dr. Awad talked about, that's in the tissue. And a clue about this came from Eileen Wren in Mark Kahn's lab that we were working with collaborating. And she noticed that in the adult mouse model of CCM, if she tried to knock out the CCM genes, she had to knock out 2 copies, by the way. That really does reinforce this 2-hit model. She couldn't get an animal with a vascular CCM unless she knocked out both the maternal and the paternal. If you want to think about it in mice terms, it would be the dam and the stud or whatever. So when she knocked out the CCM gene in the adult, you can look below. These are micro CTs by Dr. Awad's lab, and the picture of the brain on the top, no lesion. She put in one of these activating mutations that's found in PI3 kinase, which is a famous gene that's really important in growth. And sure enough, it's in cancer for sure. It's in some vascular malformations. If she put that in the mouse, in the adult, no malformation. But if she put both mutations at the same time, you see that big CCM lesion. So Dan Snelling's in my lab. So, well, let's go back and look. And we had a lot of tissue from Dr. Awad, from the Biobank of Angioma Alliance. So we just started sequencing that PI3 kinase gene in those CCMs, and at the same time sequencing the CCM genes. And that in panel A, you see that everywhere you see a black dot, that's a PI3 kinase mutation, and there's different known mutations. And sure enough, we found all these mutations in there. And if you look in the colored boxes below where there's orange and yellow and gray and this and that, we're finding somatic mutations. And many of these, we found 3 somatic mutations. These were sporadic cases where they had, boom, somatic mutation in the maternal copy, boom, somatic mutation in the paternal copy, boom, PI3 kinase mutation, and those were the aggressive lesions. And we found them in all. And that panel C there at the bottom just shows the profile of PI3 kinase mutations looks exactly like you find in cancer. Not saying that CCMs are cancer, but something was cancer's, uncontrolled growth, right? And these vascular malformations seem to be the ones with these the lesions seem to be growing. But remember, I told you that what's really important, according to Bert Vogelstein and this whole concept of the 2-hit, is that all these mutations are in the same cell. And when we do sequencing, we can't tell because we just take a glob of tissue, we grind it up, we make DNA, and we sequence the DNA, and we can find all these mutations, but are they in the same cell? And that's just a little picture there showing that what we really want to find is, what if we could sequence each individual cell? Well, that's what Dan did. We actually sequencing the nuclei, the part of the cell that has the DNA, and we did the single nucleus DNA sequencing where we take the tissue, we break it up, we do FACS sorting, then we put it in little micro droplets, and you can see there in the blue, you see everywhere where there's a round circle, that's a little droplet. And many of them, in order to make sure that only one goes in every cell, you have to dilute it so much that the average, this is a Poisson distribution for the aficionados, right? So the average is only one in every cell, one nucleus in every droplet. And then we sequence. We take all the droplets that have it and we sequence them. And sure enough, what Dan found was statistically extremely high evidence that even when we had 3 somatic mutations, the bulk of the lesion itself had the cells had when they had a mutation, they had all 3. Okay? So the idea here is that these mutations are in that same cell, and that's what's driving the lesion genesis. Okay? And so a beautiful paper came out by a group here that we have some colleagues here from China that were involved in this, is that there was a MAP3K3 also, a different gene mutated. And we missed that one. It wasn't on our sequencing panel. So what we did, by the way, we knew that this mutation was also involved in a very odd form of malformation, and has nothing to do with CCM, varicose venous malformation. So Dan went back and said, well, some of those samples where we couldn't find a mutation, and sure enough, those purple lines, those are all the vascular malformations in the biobank or Dr. Awad's that have the MAP3K3 mutation. And on the bottom, those little Venn diagrams are proving again that the mutation that's driving the lesion genesis, the cells that are driving lesion genesis have both mutations. They have the MAP3K3, and in this case, a CCM mutation, or not a CCM, a PI3 kinase mutation, okay? For the people who are gonna stay for the afternoon for the science, this point that the real important cells have all the somatic mutations in the same nucleus, in the same cell, is going to be really important for Andrew Ressler's talk. He'll say that again, but that's just an advertisement for you to stay and not go home. All right. And by the way, the MAP3K3 story makes sense. You'll see all these biochemical pathways, but the idea is that one way to create the CCM is to knock out one of the CCM gene complexes, but the other, since that complex seems to inhibit the MAP3K, sorry for the patients, the scientists can never figure out nomenclature. So MAP3K3, the protein is called MEK-K3. So I didn't do this, but that MEK3, the arrows going down, those are activating mutations. So you can either knock out the inhibitor or you can either break the brake or put a brick on the gas pedal. Either way, you can get the same CCM lesion, okay? And as we were thinking about this more, we noted that this idea of the Vogelgram, remember that there's an initial event and it's kind of like a precursor cancer, and then you get another mutation and then it's closer. And then we were thinking, many of the sporadic CCMs are associated with what's considered a benign vascular malformation called the developmental venous anomaly, or DVA, and it's very common. And there's that palm tree thing with the arrow is a picture of that. And many CCMs grow adjacent to that. And Dan and I were wondering, I wonder if that is the precursor, like in the Vogelgram. So we teamed up with Dr. Awad again, and we started to sequence CCMs and the associated DVA. And there's a lot of gibberish here, but the bottom line is that the PIK3CA mutation is in the DVA, that benign lesion, and then the CCM grows out of it. It has the PIK3CA mutation and CCM, or in this case, MAP3K3 mutation. So here's a little picture of that. The idea is you can get this CCM sporadic or familial. You have this quiescent CCM in the top case, in a familial case. And then if it gets it's a small lesion, that's not something that needs to be worried about or surgically resected. But maybe if it gets that PI3 kinase mutation, then it becomes aggressive. It becomes the more aggressive form. What we think is the opposite in the sporadic cases, mostly what you have, the PI3 kinase mutation happens first. So it drives this benign odd growth, but it's not a malformation until you get MAP3K3 mutation or CCM gene mutation, and then you get the DVA-associated CCM. So finally, and I'm thankful that the clock wasn't on because I have no idea whether I'm going over or not. I want to talk about, I'm not going to talk about any more science now. I just want to talk about, well, what other things have I learned on my odyssey? And I'm going to talk a little bit later about scientific collaboration, as Dr. Awad mentioned. But first, I want to talk about the association between my lab and what was Angioma Alliance and is now Alliance to Cure Cavernous Malformation. And Connie recognized these slides because Connie and I gave a short version of these last few slides to the Duke Cancer Center because they were wondering about how can we increase patient involvement and patient advocacy and community in cancer research. We were like, well, we got a good model here. So we gave them a talk, and I think Connie made these slides. But from my perspective, it's been unbelievable. I would not have any career or anything, and we would not accomplish any of this good science, I can tell you that, without the association, the alliance. And we, you know, there are many, many more papers, but there are actually 11 papers that would not have done if we didn't have Connie, you know, saying we can send you this tissue. And Amy was working on tissue that Connie had started with the biobank, and then later Amy sending us tissue when she was now that she's the head of the scientific branch of the alliance. We had so many contacts. We wouldn't have published that paper on the common deletion mutation and how it spread throughout America without Connie's work and Henrietta's work. And we've gotten 6 grants. So occasionally we've gotten money from the alliance, but more importantly, our association has directly led to 6 grants. And I've calculated the direct costs, which, you know, the scientists will know what that means. That's $23 million. That's a lot of leveraging, right? You know, maybe that costs $50,000 or $100,000 of patient money and you get $23 million back. I wish my stocks were doing that. But so this is, again, one of Connie's slides, but we shared with the Cancer Center. But why should we all connect? And I just took one quote from you, Connie, to share insight, information, and hope. And I think that, you know, when you boil it down to a nice fancy quote, that's it. I want to talk one more thing then about scientific collaboration. And so Issam has shown an embarrassing picture of me, and I absolutely remember that picture. I remember wearing that hat because it was freezing cold. Because my daughter's listening to this on Zoom, Dr. Awad smoked a cigar. I did not. Although I might have had that single malt, I think it was a rye and not a scotch, if I remember. He's nodding his head. Yes, and he might have had more than one. But you know, we were beginning to work together even slightly before this, and this is when Dr. Awad was in Evanston, and Bob Shankar has told me the exact date. It was in September 2009. This is the precursor of a fantastic collaboration we have now, but this is the original group. You can see Dave McDonald, who did some of that, some of the early mouse work that I didn't even talk about. He was. Did I touch anything? Is it? Oh, darn. I'm sorry. Oh, okay. Yeah. Yeah. Oh, yeah. So you can see Dr. Awad there and Dr. Mark Ginsburg, who was here. I think he's still here. And some of the people in the lab, Shang Bin is here in that picture. Some of these people are not on the team anymore because they moved on to other things. You can see I had hair at one point. And then, but that's the seed of the formation of a very successful collaboration now that we've got 2 program project grants, or I should say we've successfully renewed it. And we added to the team Dr. Mark Kahn from University of Pennsylvania. And then very recently, Miguel Lopez Ramirez, who many of you see sitting right there. And we've got this great team, but this is the genesis, you might say, of that back some time ago before we even had the full team, just sitting together, thinking about what we could do, dreaming about what we could do. And I also want to acknowledge some other people. All these people in my lab worked on CCM listed here, and some of the current people. And I mentioned Andrew's going to give a talk today that I think the scientists are going to really like. I really got a shout out to Dr. Awad, who we've had this success. I'd forgotten, I think I calculated now 30 papers together, but more importantly, we've made tremendous progress together, working together. And Dr. Kahn and Dr. Ginsburg, and somehow or other the animation didn't work. I've got my funding here, but I really wanted to thank the Alliance to Cure Cavernous Malformation. And there was a box with the CCM families, but somehow that didn't, because we do it for you, but we also have great support from you and participating in research, samples, blood, tissue, just support. So thank you.